it'll be live stream. It'll say physio on it on YouTube. Make sure it's muted. So to do this stuff, I have to put on my old people glasses. I forgot to put stuff in my wife's lunch today that she normally has in her lunch. And it makes me very sad that she's going to open up her lunch and she forgot things. But stupid empathy. I don't know why I care, but I do. Okay. All right, so let me put open the chat up here. Alt-H. Good morning. Okay, so I have the chat window up, so if someone asks a question, it'll pop up in front of my face so I can see it. Okay, so what we're gonna do really quickly today. We're basically going to talk about something that should take like 2 weeks to talk about, and we're going to talk about it in one day. So if it feels extremely overwhelming, that's probably because it is. If you want to, I mean, if you want to look at some other things about this, if I go to Canvas really quickly and get my glasses back, okay, I'm going to, this is my other class. I go find my physio class. On the home page of the physio class, there is a link to the YouTube, and I will put up my office hours and stuff up here. I thought I did that yesterday, but I guess I didn't hit save. But there's a link right here called PowerPoints. You click on this. These are the publisher generated PowerPoints that come with the book. So we're going to kind of, if the least common, the least amount of information that you need to know is know the information in these PowerPoints. So it's one of the ways to start is look through the PowerPoint and then go through class and then try to do the best you can to try to process as much of this stuff as possible. I mentioned before, you can talk to other people in the chat privately. I don't know if all my classes kind of blend together, but if you go to the chat, you can actually message individual people in the chat. And that allows you to, if you want to connect with people outside of class, that'll really help you in processing this information as things get a little bit more complicated. Right? All right.
And
so
the only person that sees that is the one you're chatting to, and then the instructor.
Yeah, I don't see it, so you can talk about crap about me all you want.
Actually, my history instructor said that that's a misconception, that people think the instructor doesn't see it, but they do.
Well, I can open up the file on. I don't see it coming up and down on my screen. Like I don't see the in the chat. I don't see people talking to each other, but I can download the chat screen from Zoom and export it as a text file, and I get a bizarre looking text file that shows the entire chat.
Oh, I see.
Yeah, so I can. I'm not going to because it's too much work, but but yeah. So technically it's not 100% private. That makes that sense, but it doesn't pop up on my screen right here, so I'm not going to see it on my screen as it goes through. But again, anything that if you have any sort of questions or criticisms or something, please let me know. I'm willing to change whatever I do, and do whatever I can to try to make people more successful. That's my goal. That's what I'm here for. Okay, and trying to remember to pack my wife's lunch properly. All right. So I've not got a text about it yet, so she probably hasn't discovered it yet. Okay. So now I know Charles knows all the answers to this because he's had his hands into one, but what's a brain made out of? This is really good stuff. Your brain is basically a bunch of skin cells with an asterisk next to it. All right. And so I want you to kind of think about that your brain is like skin. And so what color is it?
Gray.
Well, it's actually not gray, it's pinkish. It's like the color of skin. And it doesn't have any pigment in it, so it is just unpigmented skin. So if you want to think about it, it's like albino people's skin. I guess that's weird. Anyway, so when there's parts of the brain that are grayish, and there's parts of the brain that are whitish. Gray matter and white matter. Now, a lot of times people like to use the word gray matter to mean intelligence, using a lot of gray matter. Okay, so the grayish portions of our pinkish brain, okay, the gray matter, these are the neurons. The white matter are what we call glial cells. Okay, oversimplified, but we'll get more and more detail and tell you that how kind of wrong this is, but also how right this is as we go.
I'm like, did I say something or did I not?
Not yet. Not to oligodendrocytes and Schwann cells yet, to give it a chance. Okay. So there's something that's really interesting about your brain. Okay, so there are about 1 trillion cells in your brain, give or take. Okay, that's a lot. Okay, we only have about 86 billion neurons. About, give or take. And there's a really great TED talk on somebody counted the number of cells that a human has in a brain on average. You can Google that if you want. It's a very fun 15 minutes of waste of time. So she basically, everybody always says 100 billion, but that was just a guess. And the true number is closer to 86 billion neurons. Okay, so we're going to talk about the neurons first, then we'll get to the glial cells, and then I'll tell you why I was lying about the white matter being glial cells. Okay, so first of all, very simple. Let's talk about what a cell is first. Okay, so someone here has had biology, and so what's the most important part of a cell?
The nucleus.
Nucleus, good. Okay, so what's inside the nucleus? Why is it so important?
It's the DNA.
Yeah. Okay, and I should use magic sparkles for that. Let me move this because DNA is pretty neat stuff. Okay, so I'm going to put some magic sparkles and put some magic sparkle dots in here because DNA is some very exciting stuff. So DNA works in a very kind of simple but complicated way at the same time. DNA has a sequence. A, C, G, G, right? Adenine, cytosine, tyrosine, guanine, right? Biology folks, help me out here. Crickets from the bio?
Yep.
Thank you. Okay, so your DNA has these letters. Okay, and what these letters do is, again, this is a psych course, not a bio course. They get pulled out of the DNA and become RNA. And the RNA, again, oversimplified, goes out into the cell, and these little things called ribosomes. Okay, so I'm just going to draw this little ziggy-zaggy with little thing right there. That's right here. So the ribosome scoots along the DNA. So the DNA in the nucleus stays there, it's safe, keeps it protected. When it needs to send some sort of message or grow something or do something, it breaks off this little RNA. And one of these things turns into a U as opposed to another letter, which I don't remember what it is right this second. Which one becomes uracil?
Or yeah, would you ask?
Which one of these becomes a U? ACTG becomes a
Oh,
U?
the RNA. Oh, the T.
T, thank you. Just for, full disclosure, just to be 100% honest with this stuff. So, but for us, each one of these letters represents a type of kind of like a key, like a clue. And every three produces an amino acid. Again, oversimplified for psychology majors. So one of the ones that I know pretty well is if I have a CAG sequence, the CAG sequence, so CAG, this produces a amino acid called Huntingtin. So if I have one, this little Huntingtin is produced. And this Huntingtin is this little teeny protein that acts as a pair of scissors. That's the worst pair of scissors I've ever drawn. Stand by. Flat edge, curved edge. Flat edge, curved edge. This one goes to that handle. This one goes to that handle. That's a much better scissor. Okay, so if Huntington's a pair of scissors, snip, right? At the end of a sequence, and I produce this little scissors, it snips whatever protein. Following this so far? What if I have two CAG sequences in a row? What do I get?
You get a protein that's cut earlier.
There's twice as much scissors. It's cut sooner.
Right.
What if I have 100 of these in a row?
You get Huntington disease.
Huntington's disease. What happens is if we have too many scissors, too many genes that are being cut in your brain, your body slowly starts to fall apart. And so this is just an example to understand how these things work. But they discovered this because it's called Huntingtin, because some dude named Huntington or Hunter or whatever his name was, figured out this disease that was caused by genetic codes. Too many of these repetitive codes caused brain decremation. It has something to do with the way that your brain produces glutamate receptors and goes on and on. Well, that's later in the semester. But the idea of it is that the nucleus is a really important thing because it codes these different amino acids. And so it works I'm going to simplify this a little bit. These amino acids come in three basic flavors: positive, neutral, and negative. And there's some complicated ones that have lots of both, but whatever. So far, so good? So some sequences produce positively charged. Or negatively charged. So what would a zero charge be?
Neutral?
Yay, neutral. EUTRAL. Okay. So this is great. So I'm going to go back up, and this is where I wish I could do more animation with my computer. So I'm just going to draw a nice flat piece of DNA. And here's my little ribosome. And when it hits this sequence, it's going to kick out a little bit of an amino acid. And let's say that part's positive. As it moves across the DNA, by the time it gets over here, well, that's a terrible ribosome, sorry. This has a point in a minute. It'll collect all of the genes. And so it might have a positive, a positive, a positive, and then a neutral, and then a negative. By the time it gets here, it builds this thing called a protein. So as the ribosome slides along, it collects these amino acids and forms a protein. What's kind of cool is if I put a bunch of negatively charged amino acids together, they form a stick. Because the negative charge, okay, wait. Positive is attracted to
Negative.
negative. Negative is attracted to positive. What is neutral attracted to?
Either.
Say it louder.
Is it to either one?
Nope.
2 positives? Oh,
Neutrals are attracted to neutrals. So a really good way to think about this is a magnet. Magnets really like, did you ever get to play with magnets?
Yeah.
My son has these little tiny rare earth magnets. They're actually very dangerous for kids because a lot of times a kid will swallow one, which can be really bad for you to get a magnet stuck in your intestines. Anyway, but he has these little rare earth magnets that are super, super strong. We ordered them through some online thing. They looked much bigger when we ordered them than when they actually are. Anyway, but they're super strong. You cannot put them together the wrong way. You can't put the two sides together the wrong way. They repel each other. So if I put a negative amino acid next to another negative amino acid, they're going to push each other away from each other. And so if I make a big long stick of negative amino acids, it's going to form a rod. What happens if I make a stick of a negative amino acids, but then put a plus in the middle of it? What's it going to do?
Join together?
It's not going to join together. It's going to bend. Because the little plus causes this to be attracted to it, but it repels this, so it's going to bend. And if I want to make a protein that folds over on itself, try to erase with my finger, which works on a whiteboard, but not on the computer. All I have to do is put a minus and then a bunch of pluses. And this will fold over on itself. So the cool thing is by changing which flavor amino acid I have, I can make things that look like this. What if I take a protein and have a positive, 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 neutral, positive, 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 positive. So I bind those together. Or one like this. So this right here is actually something that's really important. So I take this little protein that I built. It's actually not technically a protein, but it's built by the DNA. And what this is the zeros or the neutral charges are going to be attracted to each other, but the positive end is going to be attracted to what?
A negative?
A negative charge. And it's going to repel another positive charge. So again, oversimplified, but when we have a bunch of neutrally charged amino acids, we use the word lipid, which is a fat. So what would I have here? This little structure. This little structure forms what we call a membrane. So I'm going to kind of draw these a little bit. I'm going to go back up here because I'm going to go back up to my nucleus. Wow, that went back fast. Okay. So I'm going to take this and move it over a little bit so I can, oops. So around the outside of my cell, which I now have to go ahead, I have a membrane. If I zoom in on my membrane, way in on my membrane, what I see is I see a positive, oops. I'll get better as this semester goes on. I see a little positive charge followed by a bunch of neutral charges. On the inside of the membrane, I have a positive charge with a bunch of little neutral charges. What are the neutral charges going to do with each other?
Attract.
They're going to attract. So I get, are the positives going to attract each other or are they going to repel each other?
Repel.
Repel. So what's going to happen is I have two positive charges here, one on the inside, one on the outside. And I'm going to have this membrane. Get the eraser. You just clear this little spot so you can really see this better. Neutral. And you can draw it a little bit better if you just draw a little fat sticks like this. And this one's facing the opposite way. The little fat sticks look like this. So these layers of lipid form what we call a semi-permeable membrane. Because the membrane is actually made out of layers of fat fitting in against each other. Now, is this complicated or relatively easy?
Rather simple.
Some people it's relatively simple. How about the rest of us?
It's a little complicated for non-bio majors.
Yeah, it's a little bit, it's like explaining analysis of variance at the beginning of the semester. But again, the reason why I want to say this is that I want to go back and think of the big picture here. Okay, the nucleus is a series of letters. Each three letters collects these things and makes these amino acids, or collects the amino acids. And when you put a sequence of them together, it forms some sort of protein. And the CAG protein forms a scissors. And so it trims whatever sequence of DNA is around. And so in order to make a particular protein and make a protein and how it works, they change the charge, don't change, but they select certain charges of amino acids. And when you select a certain charge of amino acid, it forms a particular shape. And that's the key takeaway, that the DNA produces these shapes of these proteins. The shape of the protein helps determine what the protein does. I took a course in neurobiology years ago, 1993. And it was so ridiculous, but even back then, they knew the DNA structure of a bunch of parts of the brain. The problem is they didn't know how it worked. They could organize the sequences, they could synthesize the amino acids, but they still didn't understand how things like we'll talk about a sodium channel worked. They didn't understand how a sodium-potassium pump worked. They knew what it was, but they couldn't figure out how it worked. And they figured it out since then, but with the better microscopes. But even though they knew what it was, they still couldn't figure out how it worked. And we spent days talking about the amino acid structure, the alpha helices of specific membrane spanning proteins. But for, I mean, again, that's a whole nother rabbit hole to go into when you get to the 500 level classes. We're at the 100 level class. So we talk about one specific one. And we talk about the one specific one that forms a membrane because it makes a lot of sense. We've got 2 layers of fat. These 2 layers of fat intertwine themselves to each other. We have a positive charged phosphate molecule on either end. So these phosphate molecules repel each other. And if I were to put another one here, the next one would try, the fat gets inside, and the next phosphate would line up against it. And if I take another one upside down here, the next one would line up against it, and the next one would line up against it. And all these little things, these phospholipid, molecules, a phosphate with a lipid, fat on it, positive and neutral charged, form this membrane. All I have to do is make another one. What does it have to do if I make another one in the DNA? Is it just going to float around there?
No,
No. What's it going to do?
gonna connect to something else.
Well, what's it going to connect to?
The same molecules.
Yeah, it's going to connect. It has to, by chemistry, it has to connect into the phospholipid. Because of the shape of it, it's going to be attracted to the fat, and because of the positive charge, it's going to be repelling the other positive charge. It's going to automatically go in and zipper itself right into where it goes. Notice I flipped it upside down because it can't just stick with its phosphate into the fat because the positive is not attracted to the fat. The fat's attracted, so it's going to spin around and stick in there. And so, again, this is very simple in kind of one way, but also extremely complicated in the other way. So I'm going to zoom back out again because there's a lot of cellular things that happen between the nucleus and this phospholipid bilayer. But why have this membrane? What's the membrane for?
Protection.
Well, protection from what?
From other things going into the cell. It's semi-permeable, so
Okay, so I'm going to write that word up here again. The word semi-permeable means that certain things go back and forth through the membrane very quickly. Other things do not go back and forth across the membrane very quickly. Okay, so things that go back and forth across the membrane very well, I'm going to make this a little bit smaller. Oxygen. Oops. Oxygen flows back and forth across the membranes pretty quickly. Glucose flows back and forth across the membranes relatively quickly. Certain amino acids goes back and forth across the membranes relatively quickly. Well, one thing that doesn't go across the membrane very quickly are ions. So again, this becomes kind of a pain, but ions, we have to add some chemistry here. Charged particles. Charged particles. Why would a charged particle not go across this membrane very quickly?
Held by the fat.
Because it's fat. What's kind of nice is like all my wires on my desk, why are they coated with plastic, I hope? Or my lovely construction wires here. Wait for it. My office is not the most accessible. I don't know if you can see that one. I can't quite get it in the camera. See that right there? Why is that? That's actually for my sprinklers. It's not any voltage. That's just to get my sprinklers from the control box to the front of the house. Why is it just a piece of wire wrapped with a bunch of tape? Why wrap it in tape?
It's isolated.
It's isolated. I don't want, when my little control box turns on, I don't want the wrong sprinklers to turn on at the wrong times. Okay. Yeah, and my wife just noticed her no food.
Aw.
Yeah. Oh yeah. Yeah. Okay. Sorry, honey. Okay. So, the idea, so the electrons flowing across the wires need to go where they need to go, and ions are these charged particles that act very much like electricity. When the ions flow back and forth, electrical current flows back and forth. Now, in these, it's electrons bouncing against other electrons in a metal, which the ions travel. Not ions, but the electrical current travels extremely well along wires. But if the current is if it's not insulated, if there's not plastic on the wires, plastic is kind of a form of fat, the signal doesn't get through as well. And if the signal doesn't get through as well, it slows down. Like this computer right here is connected to the Ethernet directly by a wire. And it's connected by a wire because it is faster than being connected through the air. Electrons in the Wi-Fi going through the air is slower than directly through a cable because of the amount of insulation and interference. And so, so when we look at this idea of this membrane, the membrane is a really important thing because it helps create this insulated unit that keeps these ions, these charged particles out, and keeps these ions and charged particles in. Hold on, I got to reply back. Just sorry. Yeah, okay. I should get another text back in just a minute. Okay, so now, one of these days I'll learn to pack her lunch correctly. I've kind of screwed up pretty much every day, a little bit differently though. Yeah, okay. So the thing about these charged particles are really important. So I'm going to draw this little periodic table here. I'm going to draw it over here. Okay, so everything over here on the periodic table, everything over this direction on the periodic table is what charge? Come on, somebody bio.
Positive.
Positive charge. Everything over here is
Negative.
negative charge. As you go down the periodic table, things get bigger. Bigger. Okay, so if I'm trying to pick on somebody who's tiny, I don't have any people that I can pick on who are tiny in this class that I know of because I can't see your whole selves, but I'm a big person. I take up a lot of space in the world, okay? And because I'm a big person, I take up a lot of space. Even someone who's very small might have exactly the same power and authority that I do. I just take up more space. So if we have something like here, which is something like sodium, it's up high on the chart. If I have something like potassium, it's low on the chart. They both have the same charge. They both have the same power. What's different?
Potassium's bigger than sodium.
Potassium just takes up more mass. So if I draw a sodium, it's here. If I draw a potassium, it's like that. So if given a standard volume, oh, it's supposed to make a little square. Okay. What could I fit more into this space? Sodiums or potassiums?
Sodium.
I can fit more sodium into the same space than I can potassiums. Okay, because sodium is small. Now, Molecularly, it's just kind of hard to visualize and hard to conceptualize. Okay, but at the same time is that in our body, in our cells, this balance of sodium and potassium is actually really important, especially in our neurons. Okay, we're going to get back to this in just a few minutes. So what I can do is I can measure the voltage. I can measure the voltage between the inside and outside of a cell. In a typical cell, in a normal cell, does the world like to have a charge? Does the world like to have things that are different charges in it? If you just let your phone sit, what happens eventually?
It dies
Why does it die?
because it has no charge.
Eventually the charge wears off. Now, in certain things like in batteries, like the little 9-volt batteries, or the one that's I'm seeing a 9-volt battery in my sprinkler thing right now, they can last a long time. Some little batteries that, like the little AA batteries, you get the Duracell batteries, you can set on a chart on a table for 10 years, and it'll still maintain a charge because the membrane that's between the positive and negatives is extremely hard and is extremely non-permeable. And if I keep that membrane non-permeable between the positive and negative, that charge stays for a long time. But on some other types of batteries, the charge dissipates extremely quickly. There's a thing called a capacitor. Never mind. Okay. So what's interesting about a biological systems are terrible as far as maintaining a charge. The semi-permeable membranes are, these biological organisms don't maintain a charge very long at all. And so as a result, the voltage across a membrane, if I wanted to create a charge, only lasts a short period of time. And so your body uses a tremendous amount of something, which is the next thing we have to learn here, which I'm going to draw up here. Your body uses a tremendous amount of this. So adenosine is a molecule, and I stick a phosphate onto it.
3, actually.
3 of them, yes. That's why I drew 3. And when I stick a phosphate onto it, it gives this power. It's like charging a battery. It's a chemical charge. When I break a phosphate off, it releases that power. Okay, I've never really quite understood how this works chemistry-wise, so I've just kind of accepted it. It never got past the chemistry where I understand how it's made, and I understand that it does, but I don't understand exactly how it works in reality because I can't wrap my head around it. But
Do you want me to tell you?
no, because then I'll forget things more, and I will forget what we're having for dinner
It's
tonight.
pretty easy.
It's complicated. It's chemistry.
Got it.
I just accept it.
It's okay.
One of those
it's okay.
things you can just say, yes. Yes, it does. Okay, so what happens is adenosine triphosphate becomes adenosine diphosphate, and now it has 2, which then it becomes AMP, and it has, well, 1. Every time we use this ATP molecule, we break off these phosphates, it becomes smaller. And what's really neat, I think, and this is part that makes me happy, is that then there's a neurochemical called adenosine. And adenosine makes you feel sleepy. Why would you feel sleepy? With adenosine. Or you can drink coffee and block the feelings of adenosine.
Okay, I have a question.
Sure.
So you're saying the ATP, okay, loses a phos, and then it becomes like ADP, and then it can become an ANP.
So with the skepticism in your voice, that's how I learned it. But that's so when we start looking at when you have to repair these adenosine molecules, I might be wrong on that, but that's how I learned it a long time ago. So that's how I keep broadcasting this. And so I could be wrong.
Okay, interesting.
Great. Thanks, Charles. I'll have to go read some more stuff again. But that's this is how I've always understood it. And that how do you get your ATP back together? Again, following the logic that I have in my head, which might be wrong. How do you repair these? How do you make ATP? There's these little parts of your cell called mitochondria. And the mitochondria create or build ATP, glucose, oxygen, and water. What is the byproduct of all this?
Energy.
Yeah, well, that is the ATP. It rebinds the phosphates back together. One of my roommates when I was in college was a bio major, and he had the entire cycle he wrote on the wall of our apartment in Sharpie. Turns out it takes a lot of base coats to get rid of Sharpie when we tried to move out. Anyway, put paper up next time if you're going to do that in your apartment wall. He had that, and then he had the two other cycles and how things create energy. But so your body is constantly using ATP, and it's constantly rebuilding ATP. And to me, it's interesting is that adenosine in your brain stem is used to inhibit cells. When you start getting tired and feeling tired, there's more adenosine building up in certain parts of your brain. Now I have to go rethink how I imagine this for how many years, but that's okay. That's what Charles is here for. Okay.
Sorry.
No, hey, reality is much better than mine.
Does it talk about this in our book?
Yeah, it doesn't go into too much detail about this. That's why I don't know as much detail about this. But the idea is that the mitochondria are the ones that are producing ATP. The reason why we need this ATP is to create a charge. Whoa, sorry. Is to create a charge. So ATP is energy. And what the energy does is it expels sodium out of the cell. Laundry's about to go through a spin cycle, sorry. And it puts in potassium into the cell. Expelling sodium, bringing in potassium. Expelling sodium, bringing in potassium. Okay, so we're going to come back to that in a little bit. So we're going to talk a little bit more about neurons.
So does adenosine slow down the activity of the neuron?
So I don't know if adenosine itself slows down the activity of an individual neuron. I know that in parts of the basal forebrain, and where your brain pays attention and your brain focuses on your short-term memory and deals with processes going around you, that if you have high levels of adenosine in that part of your brain, your attention span goes down. You actually focus on being tired if that part of your brain has too much adenosine in it. And so I think it's a nice metaphor of understanding how ATP works. I don't want to say that that's how it works, that you build up too much adenosine by breaking down too many phosphates, just because that biochemically that probably doesn't work, but it makes sense in understanding adenosine and understanding the idea that this ATP molecule, you use it constantly. You're constantly using ATP, and the ATP is actually really important for keeping our cells functioning. Some cells can work okay without ATP, but most cells need tons of it, especially your neurons. Your neurons use a tremendous amount of this ATP, and they need to be constantly cleaned and flushed and everything else, but we'll get there in a minute. Okay. So neurons have very unique shapes. They're the same as your skin cells, except they have some very unique characteristics. Okay? They have these things called dendrites. These dendrites are like extensions of membranes. Okay, so I'm going to zoom in on a dendrite. So I'm going to make this smaller so we can kind of see what it looks like. Oh, I think our pool guy's here. That's Nani's pool guy bark, just so you know. Okay. So the people in methods last year, you always knew when the pool guy came because the dog was barking. Okay. So dendrites have membranes, just like the cell. But they have these really little things, small things on them called spines. What the spines do is the spines grow all over the dendrites, and they grow all over the dendrites where another neuron is coming close to them. And the spines form this thing called a synapse. So what forms a spine is another neuron getting close to them. And so these dendritic spines are really important. So it's kind of easy to think if we zoom out, well, that information just comes in the dendrite. But is that actually true? What does the information do?
It travels along.
It travels along the dendrite. Information can come in anywhere along the cell. And technically, anywhere that another neuron is crossing a cell, a spine can grow. And these little spines are where the connections are. Dendrites are really fascinating. They grow. There's these little fibers called microtubules that if a neuron is getting a lot of information from one side, if there's a lot of information coming here, the dendrite will grow and grow more spines out toward whatever information's coming in. If this dendrite's not getting any information, it'll get pruned. It'll prune right off. This is happening in my poor son's brain right now. My son, as he goes through these growth spurts, he grows a tremendous amount of new cells. And then all of a sudden, all the dendrites start growing. But they're not connected to anything yet. And that's why these little creatures, they go through these growth spurts. Babies will do this. They'll go through a period of time where they're extremely cranky and overwhelmed by everything. And all of a sudden, they kind of get up in the morning, they look around and like, oh, I understand the world in a different way now. It happens so fast with babies. As people get older, this happens slower. And that's why for me, like I remember the ADP thing, whether it's actually exactly how I remembered it or not, but I remembered it the same way for so many years, that seems like the right answer. Whether it actually is the right answer or not, those dendrites have formed what we call a pathway. And that pathway becomes the way things are. It's the same thing as if you learn how to stand. One of my students years ago had a limp. I'm like, oh, what happened to you with you? She goes, oh, I sprained my ankle about 5 years ago. I'm like, wow, and it still hurts? She goes, no, I just learned to walk this way.
Yeah.
But why did she learn to walk with a limp?
Became her normal.
It became her pathway. The dendrites have formed that pathway, and she just kept walking that way. I had to relearn how to stand. I used to stand with my back a little bit bent and my abs squishy. So when I stand now, I'm flexing my not abs, I don't have abs, I have one. But I keep my stomach tense and my lower back a little bit tense when I stand. And as a result, my back doesn't hurt and my feet don't hurt as much because I'm not slouching and disproportionately putting my feet on down. I don't like to sit all day. That's why I'm standing now. And because if I sit all day, my back slowly rounds and I get archy and then it hurts my back. And then my legs hurt because I'm not moving. But again, so learning, that's a long way down the path from where we are right here. But these dendrites really do shape the way that our cells interact. They also, the axons. The axons also shape how we interact because they do the same thing. The axons send out information and they form end bulbs, or axon termini, or presynaptic terminals. So I like the word terminal. Terminal sounds good. Terminals and spines, but these end bulbs, these axons produce these end bulbs that look just like the spines, and that's what connects. If an end bulb is growing next to a dendrite, a spine will grow where that is. If the two cells start communicating and they become friends, I don't know how to say that properly, but if that synapse is useful, then that spine grows and the dendrite gets thicker in that particular area. So the axons they have what we call sprouts. The axon can sprout out. If an axon is sending a lot of information, oftentimes it'll form sprouts, collateral sprouting. And the sprouts form end bulbs that connect to the other cells around the particular cell that they're connecting, or more parts on the same cell. What's interesting, and this is mostly true, all the cells have one origin of an axon. There are some types of neurons called microneurons or interneurons that can have multiple sites of axons. But from the way that it generally works is we have one axon start. It's called the axon hillock. Okay, so the axon hillock is a bump on this cell that causes the axon to grow. And when I say causes, it's because the axon hillock is what stimulates the axon to grow. If the axon isn't used, that axon will fade and not grow until something else causes that axon to grow. We'll talk about this later in the semester, but there's a disease called Alzheimer's disease. Alzheimer's disease is where you get these tangles that clog synapses. And if these tangles clog synapses, what happens with the axons? The axons stop sending signals. They stop sending out looking for dendrites. And when they stop sending out looking for dendrites, the axons then wither and they stop connecting to other cells. So even though if this one is clogged, these are still connected to other things, you're good to go. But what if you have only one connection and it gets clogged? What happens to the whole cell?
It dies?
It dies. Something that's interesting about this ATP that we talked about is that if your cell isn't being used, what happens to the amount of ATP it has?
Comes less.
Comes less. Now I'm going to have bug trials on this one again, but I know that on days that I don't work out, I feel more tired all day. I don't know if it's psychological or if it's ATP, but when I exercise, I have much more energy all day. And it could be just psychological, but it also could be the fact that if I use a bunch of energy first thing in the morning, then I rebuild that energy, I seem to have more energy throughout the day because my body is trying to reproduce or produce more ATP. On weeks that I work out a lot, I'm physically tired, but I have more energy. The more you work out, the more your body gets better at producing energy and reproducing this. I don't know how about the science of that, but that's what I've been told by my fitness folks for years. The more you burn energy, the better you are at making energy. So again, so So the center part of the cell is called the soma. And the center part of the cell is everything that we just talked about a few minutes ago with the DNA, and it's got the mitochondria in there. There's mitochondria throughout the entire system. There's membranes throughout the entire system, but the soma is where the DNA is. Even if I have a catastrophic failure of dendrites, like I slice my finger off, or right now I have a bad heel spur on the bottom of my foot, and that causes the nerve that goes to my heel to be impinged because the tissue around it's swelling up and it's pinching off a nerve in my foot. So if I damage that nerve, that means my foot's done forever. I will never feel that part of my foot again.
No.
No, because what I can do is once I get it fixed, I had it fixed for a while, then I broke it again. But once I get it fixed, don't run and chase Frisbees barefoot, by the way, when you're old. Just saying. I've hurt myself catching Frisbees like more times than pretty much anything else. First time I pulled my calf, catching a Frisbee. Maybe I should play with somebody who knows how to throw one. Yeah. Never mind. Okay. But, and this time, actually this last time I hurt myself, I stepped in a hole at the beach while playing Frisbee. So it's fine. I'll get better. So, but I can damage my axons and I can damage my dendrites. As long as I keep the cell body intact, eventually I can regrow some of the connections. If you sever a really major bunch of axons or a large number of dendrites, it takes a lot longer for those to recover. I hurt my back. I had a slipped disc and I lost the feeling in my left leg for a while. And it was kind of terrifying. So like, okay, is this always going to be numb or not? But it turns out that over the course of about, I don't know, 2 or 3 months, the feeling came back and I regenerated the connections of the nerves back there as the swelling in the area and the impingement that was occurring went away. The rest of the neurons kind of came back. I still have a few dead spots on my leg where the nerves haven't regrown yet, but over the course, probably in a couple of years, they'll be back as long as I stay active, I think, I hope, anyway. Okay, but what if you actually do damage the somas? What happens?
It doesn't repair.
You can't repair. But what you can do is activate the cells around it to replace the functions of that particular cell. And that takes a lot of occupational and behavioral therapy to help people replace individual cells. One of my neighbors had a major stroke and she was doing extremely well getting her language back. And then her and her partner went on a cruise. And when she came back from her cruise, she had lost all of her language. And it was because she wasn't practicing. And so she went back to her old pathways, and her old pathways, the ones that have been reinforced, walking with a limp, if you want to use that metaphor, those old pathways don't exist anymore. So the new pathways weren't reinforced and the new pathways didn't strengthen. When she was doing her therapy every single day, her language got better and better and better. But she was probably drinking alcohol on the cruise as well. I didn't ask, but I'm going to guess that was a thing to do. And so what happens when you drink alcohol?
Discourages the growth of new pathways.
Yeah, it discourages the activation of cells, not just the growth of new pathways. It discourages the activation of cells. Cells slow down. Cells slow down, the activity slows down, the growth slows down. I worked at a research lab where we studied fetal alcohol syndrome back in college. And we studied the rat's brain growth during periods of exposure to alcohol. And you could see a huge decrement in dendritic sprouting during the time that alcohol was administered. Rats' brains are very predictable of when they're growing. So at postnatal day 14, the occipital lobe was growing for the rat. And so if we exposed them to alcohol on that day, we could make rats that couldn't understand their physical environment nearly as well. They did bad at tasks like that. I think it was postnatal day 19 or 20, we could mess with their cerebellums. and that their cerebellum would be completely non-innervated, which we'll talk about later in the semester. But so by very specific times when we know that the brain is growing, deficits can cause problems in the specific sprouting. And for the poor rats, we would slice their brains up and look at their brains to look at the particular parts of the sprouting that was going on. Yes. My soul will be sliced for many years by rats in purgatory because of that. So now.
And now they're at your house.
Now they're invading my space and slowly taunting me. Hey, remember my cousin Vinny who worked at the lab? Yeah, okay. This is from him. I'm going to eat your tomatoes. Yeah, they ate all the almost ripe tomatoes they would eat. They wouldn't eat the ripe ones. They wouldn't eat the green ones. They'd wait till they're almost ripe. So you're like excited that you're going to have a tomato. Like, no, I'm going to eat that one. And just eat half of it. And just lick the other half and look at you. Yeah. Stupid human. Okay. And they won't touch my peanut butter traps anymore. So I got to figure out something else. Maybe stop trying to grow a garden. They don't eat the cucumbers though. We like cucumbers. We eat those a lot. Okay. Whew. Is this overwhelming or okay so far?
Overwhelming.
Overwhelming. Okay. So again, and Charles can tell you this, there's so much more detail that we could go into that we're not.
Yes, that's very true. And I'm still trying to figure out what did we learn today? Can you do a recap, because I'm really lost. I'm trying to see what you're trying to get to.
Okay, let's go back to the beginning. Okay, let's go back to the beginning. So a standard cell, we have 86 billion of these. Okay, standard cell. There's a nucleus in the cell, and the nucleus generates these proteins. And the proteins are made in a specific shape. The shape of the protein determines what they do. Okay, so we talk about the membrane, and the membrane is a very specific shaped molecule, and the molecule forms this kind of semi-permeable bag. So far, so good?
Got it.
Got it. So the membrane surrounds the nucleus, and it also forms things called dendrites. It also forms things called axons. It forms things called end bulbs. The reason why the membrane is semi-permeable, it allows some things to flow back and forth across it very nicely, like oxygen and water and fuel, amino acids. But some things it doesn't allow to go back and forth across. So ions, these charged particles, don't flow back and forth across very easily. And there's a reason for that, which we'll probably talk about next class. We'll talk about the action potential next class. But the way this works is that this cell forms a very specific shape. And if the cell needs to grow more neurons, grow more membrane, the DNA just starts producing more of this membrane. So if, for example, let's go back up to my end bulb up here. Okay, let's say there's an axon coming in from another cell. There's an axon coming in from another cell. And this axon is releasing a chemical. We'll talk about those later. What is the dendrite going to do?
Can you say that again?
So an axon is coming in from another cell. So this blue neuron is coming in from the side. And we have all these things that we've talked about so far. We have these phospholipid molecules are being made. And so this axon is coming in and releasing a chemical. What is the dendrite going to do with this chemical?
Receive it?
But what's it going to do? How's it going to react to it? Here's the word right here.
Oh.
It's going to grow membrane. Why would it grow a membrane?
So the spines are like specific parts of the dendrite, of the bulb?
They're really just kind of growths on it's just like a membrane kind of bulges out and grows toward this axon coming in, saying, ooh, hey, you're cool. I like the way that you talk.
Okay.
Okay, swipe right or left, I don't know. Okay, so modern metaphor. So all of a sudden, we start producing more membrane in that particular area. So the cell receives this chemical from the axon and says, hey, that sounds like a good idea, and starts growing and forms this spine. And if another axon is coming in from over here, what's going to happen at this area of the dendrite, up where the yellow one is?
Another membrane.
More membrane is going to start to grow. But if this part up here, by the magic pixie dust there, okay, if this part right here grows, I mean, is not being innervated by an axon, what's going to happen to it?
It'll get pruned.
It'll get pruned. And so maybe the neuron will produce this Huntington chemical. It's not how it works, but it's just kind of a metaphor. But maybe it'll just produce a pair of scissors and just cut out this. I don't want to cut the whole thing. Put it here. And so maybe it'll just say, you know what? I don't need this piece of dendrite anymore because I'm not being used. And so what happens is that so if we go back to the beginning of this, inside the DNA, it's producing these messages. And these messages are going out and flooding into the cell. They're taking feedback from the interactions that they're getting from other cells. And the interaction that they're getting from the other cells produces the shape of these particular neurons. So there's all sorts, neurons come in lots and lots of different shapes. So again, consolidating that into like 2 minutes, is that a lot easier to understand than trying to go through all the little tiny details?
Both together.
Okay, so thank you, Charles, for asking that, because it is kind of convoluted, but I wanted to introduce you to a bunch of terms. I wanted to introduce you to ATP. I wanted to introduce you to these amino acid charges, because it's going to make sense next time. So every time we say it, every time we say some of this stuff, I'll front load with you some things, talk about some stuff, and then when we come back, we'll go through them again, and then all of a sudden they start becoming used in more specific context. Like AMP, for example. We're going to talk a lot about what's called cyclic AMP. And so by introducing the word AMP, adenosine monophosphate, to you, later when we talk about cyclic AMP, it's familiar. You already have some sort of neural interaction for that, and then we add more to it afterwards. So at the beginning of this class, it's just like, but then as we go, it starts making more sense. That's why I know, and it's very fun, but at the same time, extremely difficult, because you have to know so much vocabulary just to understand how one cell works. And there's 86 billion of them. And that's not even including all the glial cells.
That's true.
Yeah, okay, so getting back to this. So the DNA produces these membranes when the neurons are talking to each other, when they're releasing chemicals. What happens is the chemicals cause the DNA to produce more membrane. We call these spines. We also call these end bulbs, depending on if they're sending or receiving information. But they're basically the same thing. The membrane swells in a particular area, and when the membrane swells in a particular area, then it encapsulates and it allows the cells to connect to each other a lot better. Okay, so now I'm going to draw a different kind of neuron. This is what we call your standard NIH neuron. Okay, it's your basic neuron. Okay, so I'm going to draw one that looks like this. And there's a ton of really good pictures that you can find of different types of neurons, but I'm going to draw one that looks like this.
Multipolar.
Yeah, and this is, well, we use the word, yeah, pyramidal cells. So we have one axon. Okay, sorry, it looks like a spider. Okay, one little tiny axon with 1 million inputs into it. And not 1 million, but some of them have over 10,000 inputs into them. Or 10 DDO, whatever the hell that means. 10,000 different axons or dendrites coming into them. So what would this cell do?
Process.
One axon.
It processes information and then sends a message.
Well, the way it processes information, it takes input from lots of places. Before it sends a signal, it gets input from lots of places. These are located in the center of your brain that help you move. So how can I scratch my nose? What do I need to know to scratch my nose? That was Wally, I couldn't hear it. So think about, what do you need to know to scratch your nose?
How far your hand is.
Well, yeah, well, where your hand is to start with, whether you're holding your drink or not, whether you're holding a pen in your hand or not, whether your dog poo bag just broke or not, which is an awful, that happened last night actually. I didn't get poo on myself, but the bag ripped as I was scooping it up. So I had to like fold it over and it was awful. Okay, but does that matter whether you scratch your nose or not?
Yes.
Yes, thank you. No, not at all. I don't care. Yeah, it's good. Okay. So the idea of all of this, there's so much information that has to come in. Like when I say scratch your nose, do you have to figure out exactly where in your nose is itchy?
Yes.
Yeah, that all of that information has to go in to control just the neural connections to just the itchy part of your finger, because you don't want to start scratching and then get to your nose. You get to your nose and then scratch. So you have to control and manipulate all of these variables, and that's what these pyramidal cells are for. We have other neurons that are not nearly as complicated, and they're much, much, much easier. They're called unipolar neurons. These are sensory neurons. So this is the neuron that's in your nose that's itchy. It's sending a signal. There's not much processing going on. Signal gets sent, brain feels itchy. Not much going on. We can also talk about what we call bipolar cells that look like this.
Yep.
Okay, so we have these cells that are hugely integrated, that funnel and sort information. A lot of these cells there's the type of cells called a Purkinje fiber, which is in your cerebellum, that organize a tremendous amount of information. They all run through stop signals, which is cool. We'll talk about that later. But these unipolar and bipolar neurons, we have these little tiny neurons that look like this, which they don't really have any axons. They don't really have any dendrites. Hi.
Caleb won't get out of bed.
What time is it?
11.
It's 11. It's 10:49.
Yeah.
Are you getting Lucas and Claire?
No, we're going to tomorrow.
Oh, okay.
But he won't get out of bed.
Wait, we're cleaning the house tomorrow.
We are?
Lights
Andrea's
up.
coming.
Andrea's not here. Their family, they have COVID, so they can't come.
They have COVID?
Yeah, we're cleaning the house.
Well, we're going to the beach, so you're cleaning the house.
I'm cleaning the house. They're going to the beach. Okay, go away. My 14-year-old won't wake up. Okay. Well, he's awake, but he just won't get out of bed because he's 14. Okay, so these little guys are neuromodulators. They don't receive signals. Well, they do, and they don't really send signals, but they do. We call them microneurons or interneurons. They're kind of influencers. They sit in between the other cells, and they release chemicals, some at times and some in response to other chemicals. They don't necessarily have specific dendrites or any specific axons. It really changes the way we think about the brain when they discovered these about 15 years ago. They didn't really know they existed because no one could measure them. And then somebody with a better microscope at UCLA saw them and said, hey, what are those? And then it led to the discovery of these little tiny things. Whew. All right. So now we go back to our standard neuron here. Okay, so what kind of neuron did I draw now? Was the original standard neuron?
Multipolar?
Yeah,
Or
it's a multipolar neuron.
parameter?
Okay. And so because it has multiple dendrites coming out, I didn't draw the bipolar one 100% correctly. The bipolar one should have been drawn like this. Sorry. Okay, so the bipolar neuron has dendrites, soma, axon. But so the nice thing about these is they all do the same basic thing. They all take input and all have output. Even these little tiny microneurons have input and output. They're all basically the same. They all basically work the same way, except for the microneurons. The microneurons, I don't know specifically how they work well yet. The literature that's on them is beyond my ability to understand reading it. It's a lot of neurochemistry stuff that I just don't understand. Okay, so Zen moment here. We have 86 billion of these. Most neurons talk to other neurons. The input comes from other neurons. The output goes to other neurons. And they form what we really like to call a neural network. Okay, so you're going to be talking about you're going to hear me talking a lot about neural networks, about how these basic neural networks work. And the neural networks work by the connections between cells. After next class, and we're going to talk about the synapse and how the synapse works. But after those synapses, we're not going to talk about individual cells anymore. We only talk about individual cells for the first small part of this, for this class, because we're much more interested in the neural network, not in individual cells. A lot of psych majors ask, well, why do I need to know how a single cell in my brain works? If we want to try to answer that, why do you need to know this as a psych major?
Your brain is made up.
This is the foundation of your brain. This is what your brain's made from. And understanding how amino acids form these things called membrane spanning proteins, which we're going to talk about next class, is not hugely important in your grand scheme of understanding things. But understanding that the sequence of DNA produced the shapes of these things does help. How do you think they engineer drugs? What do they think they do to find new drugs?
I know that they make them so that to certain cells, right?
So in order to make certain drugs, well, the way they used to do it was just go find strange plant compounds and have people take them. But how do we know which compounds make a difference? How do we know which compounds work? Why are drugs that look like a neurotransmitter serotonin psychedelic?
Because they replicate what's already in the brain?
They have the same physical shape as other chemicals that are in the brain. Hey, he's awake. Look at that. Coming out for some high fructose corn syrup. Good job, buddy. I put them in the fridge for you, by the way. You're welcome. You're welcome. So by knowing how things are shaped, you can then understand how they work. So a lot of the research with trying to make a COVID vaccine, one of the first things that they had to do was figure out what shape this thing was. What does it look like? how can I make an antibody that will attack it and not attack other things in the cells? Like injecting the bleach thing just doesn't work.
No.
Sorry. I'm glad nobody in leadership would suggest that, because that's
Really? I thought it did.
no. stop freebasing your bleach, Charles. Well, I drink hand sanitizer every day. It's just
They had Kirk capsules too.
yes. And I'm drinking tonic water, but I'm using hydroxyquinine as opposed to the stuff that's in tonic water. Okay, anyway. Hydroxychloroquine, whatever the hell it is. But so as you start to really understand these individual cells, just something like, we're just going to just do this. This is how I want to finish today, because just to show you why this is important that you need to know. Alcohol. Okay. Is alcohol good for you or bad for you?
Bad.
Good if you only have one.
No, it's still not good for you even if you have one. Alcohol is a toxin. Okay, and sometimes your life is more toxic than the toxin that alcohol has, so alcohol can be beneficial for some people, comma. It doesn't help your body at all. And sorry, Charles, but even the red wine from wonderful France without the sulfates in it is still not good for you.
I know.
Okay, but it's a glorious thing. Don't get me wrong. It causes lots of other positive benefits in the consciousness, but it's still, alcohol works in a very simple way.
I'll survive with a glass of wine. I'm fine.
Yeah, it allows chlorine into the cell. Wait a second. So drinking chlorine bleach, drinking is the cure for COVID. Actually, turns out people who are drinking are more likely to get sick than people who aren't. Shoot, so much for my bleach theory. Okay, so if I allow chlorine into a cell, this is something that it goes across a type of channel, a membrane spanning protein that's produced by something called a GABA receptor complex, which is a series of amino acids formed by alpha helices of fill in chemistry here. Okay, but if I allow chlorine into the cell, the cell slows down. Why is this sometimes beneficial and positive in human experience?
Help you feel calm?
It helps you, quote, feel calm. It actually doesn't make you any calmer, but it changes your perception of being anxious. By changing your perception of feeling anxious, it changes your anxiety. The fun part about it is your anxiety still present, even under the influence of alcohol?
Yeah.
Yeah. And we all know this, because if you're really anxious and you drink so you're not anxious and you go to sleep, what happens at 2 a.m?
You're anxious again.
And it rebounds, and you're actually more anxious than you were before you went to sleep. Yay.
And that's why
That's why you have to just keep drinking. No, it's not the healthy response to it. We like, it's just, yeah. And well, sadly, that's how my dad passed away. He just decided that to keep drinking in order to stop feeling anxiety. And that's why he's no longer with us. He died when he was 52 from that. Because alcohol slows down all cells, not just the ones that help you stop thinking. It slows down the cells of your liver. It slows down the cells of your stomach. It slows down the cells of your skin. It makes you age faster. It's just not a good substance in the long run of self. That's why small amounts are not horrible, but large amounts can cause, well, we'll see. I'm still kicking, so because I don't drink very much anymore. I try to avoid as much as I can, aside from the 5 or 6 a day. No, I was kidding. So now, so again, so this understanding how alcohol works, it works on the cellular level. Individual cells is affected by alcohol. So even though you talk about this in terms of how, why do we need to know this? A lot of the things in our day-to-day existence function on each individual cell and work on each individual cell of your consciousness. So, okay, let's zoom out all the way out here. How important is it to you that you know amino acids and their charges and that kind of stuff?
Important. I guess it's relative at that point.
For you, for most of you in this class, it's not that important. The reason why I talk about it is to give people a reason why something occurs. People love to ask why.
More wanted.
Say, you're gurgly, I'm sorry. The reason why it's important is because if you understand that there is a code that produces the shape of these things, it's much easier to understand that these things have different shapes. And when we talk about the different types of channels next time, I want you to remember that they have different shapes. So we talk about a sodium channel, we'll talk about a potassium channel. They're made by a different sequence of RNA. The different sequence of RNA makes them a different shape so that they can do something else. The charges on the periodic table, I'm not going to ask you to memorize the periodic table. Just know that potassium's bigger than sodium. Chlorine is negative, sodium is positive. The basic underlying thing of this, the idea that adenosine triphosphate, when you break these adenosines off, they form energy. If I need to put the energy back, I need to use glucose and water and oxygen to stick the phosphates back on my adenosine molecules. And that our cells are constantly growing. What allows the cell to constantly grow? ATP. As long as the cell is getting fuel, the cell will continue to keep growing. And the cell, the most important thing that causes the cell to grow is, it's right here in green, and it's very tiny. What causes a cell to grow?
The membrane.
But what causes the membrane to grow?
Another neuron.
Connections. Synapses, connections. So what causes cells to grow? Whoops. What causes a cell to grow is connections. So a cell that's in the center of the brain that controls movement is going to have a tremendous amount of connections, because that cell is getting input from lots of different areas in order to produce movement. But a cell that's in your sensory system, it gets information from one sensory nerve. So it doesn't have a lot of information. And it really doesn't have a lot of thinking to do either. Because all it basically says is, well, I feel something on my finger. That means that there's something on my finger. It doesn't really have to do with a lot of very complicated things. One of my students, a bunch of years ago, may have been under the influence of a little bit of extra chlorine, and he punched a window. Don't punch windows. We want to guess why?
Cut yourself?
He cut himself. So on his hand, he cut the nerve that goes right through here. The nerve that runs, Charles' name?
The palmar nerve?
Yeah, the one that connects your fingers to your...
The digit 4 and 5?
Yeah. So he cut this.
Oh my God.
And so the doctors sewed it back together. Well, they used super glue or something. I'm not sure exactly how. You just kind of stick them back together.
Yeah.
The problem with that is all of a sudden where one finger should be in his brain is now connected to somewhere else in his brain. Because did they sew the nerves back together exactly how they were?
No.
No, that takes a lot more skill and they can't really do that. But actually it doesn't really matter because all he had to do was sit and rub his hand like this and bend while looking at it and bend his fingers and move and touch his hand. And what did his brain figure out in about a month?
Rearrange.
It rearranged. So like this part of his finger felt like over here and this part felt like over here and this part, it was all messed up. But by rubbing his hand and moving his fingers, his brain goes, oh, I get it. That's this, that's this. Okay, we'll resort it out. And what it did is it just reconnected these dendrites to different areas of his cortex so he could start to feel again. That's what happened with my leg when I lost the feeling in my leg. It felt like my leg was really weak. But when the doctor put his weight on my leg, he goes, okay, kick. And I launched him. I'm like, oh, I guess that is working. You have a sneaking child about to attack you, Anna. Watch out. Can you sign this, Mom? Okay, so, but again, the more we talk about this stuff, the more it makes sense. And the more you start to understand it, the more it makes sense. And as you're going through and trying to figure out exactly what we're talking about, we'll keep adding more and more layers to this. And eventually, we're going to build what's called a neural network. We're going to be not talking about individual cells anymore. We're going to be talking about clusters of cells and start talking about pathways of cells. And stop talking about individual transmitters, but talking about actual ways that information goes from part A to part B. But in order to do that, we have to go through the boring talking about this stuff first. Okay, so should you go look at that slideshow that's in the, not the slideshow, but the PowerPoint thing that's on Canvas? When we go through the Canvas slideshow, look at the PowerPoint that the textbook puts out. See how is it different? Is it the same? Is it, how does it relate? And then, so hopefully next class when we come back, we can go through this process again and have you draw some stuff. And nobody draw junk on the screen, please. But I mean, you can if you want to, but whatever. Waiting for Zoom bombing. Okay. Oh, Summer has a cat. It looks like a skeptical cat. Aw, and there you go. Thank you. Can I do that on my computer here? My laptop won't let me annotate. Annotate. Oh, it does. I'm drawing a fly.
Oh. What is that?
It's a fly. Okay. Actually, I'm drawing in brown so I can just make a turd. There you go. Okay. So now, if you have questions or anything else, I have my office hours. I'm going to go back to here really quickly. Glasses back on.